And I'll now introduce our first speaker, Mike Ryan, to come and talk to us about mitochondrial biology. Thanks, Mike.
Okay, so thanks very much, John. I'm sure, hopefully we can see everything with the lights like this. So as John alluded, I'm going to talk to you today about a general introduction of mitochondria and mitochondrial function. And this is a picture I often have at the beginning of my slides showing a mitochondrial network sitting within a cultured cell. There's a nucleus here, and these mitochondria radiate around and outside of the out towards the cell periphery. And you may often see mitochondria in a different light, maybe something with an outer membrane and an inner membrane and cristae, something like that. And if we look at this, this is basically the general structure of what we see in textbooks. Now, mitochondria themselves are really complicated beasts. We all know that they are very important for the production of the bulk of our cellular ATP, about 90% in general organs. We turn over almost a body weight's worth of ATP a day. Now, this is an absolutely critical function of mitochondria, but we do have many other functions within mitochondria that are important. Think, for example, production of heme, iron-sulfur clusters, steroid hormone production, fatty acid synthesis, many other processes occurring there as well. And likewise, mitochondria are involved in various signaling processes, and there's a huge amount of research going on in the signaling in terms of calcium signaling, production of reactive oxygen species, even as mitochondria is a platform for innate immunity events. And of course, what's been studied very strongly over the last 15, 20 years is apoptosis and programmed cell death and the role of mitochondria in that process as well. So getting towards this function, we have obviously mitochondria that can cause, where we have a lack of mitochondrial function, we can cause classic mitochondrial diseases where we've lost the ability to produce enough energy, and that can then arise with defects in various tissues, and of course create lactic acidosis is a common event, and neurological defects and muscle defects as well. But mitochondria are also implicated in other elements, including aging, neurodegenerative disorders, and also common diseases, including Parkinson's and dementia and cancer. So when we look at mitochondria and start looking at the proteome inside, mitochondria contain about 1,200 proteins. So it's about 10% of the total cellular proteome go into mitochondria. The most recent mitochondria database coming from Vamsi Mutha's database characterizes that there are currently 1,158 different proteins within mitochondria, and our organs contain maybe about 75% of proteins that are common of this complement that are there. Now, something that needs to be appreciated, and I think most of the audience here understand this, is that about 99% of the proteome actually are encoded by the nuclear genome. The proteins are actually made outside the mitochondria, and they're imported into the mitochondria where they go to their respective subcellular compartments. The other 1% is encoded by the mitochondrial DNA, which obviously is a very big topic of today's meeting. It encodes in humans 13 proteins. These are all proteins of the respiratory chain, all core subunits of that, but we need many other subunits that are encoded by the nuclear genes to come in to integrate with these 13 polypeptides in order to produce a competent and functional respiratory chain to generate ATP. With the mitochondrial DNA, we also have other genes involved that are involved in making these 13 polypeptides, including 22 tRNA genes and 2 ribosomal RNA genes. Even the ribosomal subunits that are involved in bringing these ribosomal RNA genes together to transcribe the RNA into the 13 polypeptides are encoded by the nuclear genes as well and need to be imported into mitochondria. So when we're looking at mitochondrial disease, and this is a nice figure from Vamsi Mutha's one of the recent publications and reviews, basically indicates that there are really huge numbers of different symptoms and organ defects that can arise with defects in mitochondria. So we estimated there's about over 150 distinct genetic mitochondrial syndromes affecting many different processes, including muscle being a very common element, peripheral neuropathy, stroke, ataxia, deafness, optic neuropathy, and the like. And we'll hear a lot more about this in later on. Now, a large subset, very many of the mitochondrial defects are related to defects in oxidative phosphorylation. So this is involved in the production of ATP, for the energy for our organs and cells. And there's so far around about mutations in over 120 nuclear genes encoding mitochondrial proteins that cause mitochondrial disease, and 35 of the 37 mitochondrial genes cause disease as well. So I'd like to go through a little bit of this just in basic detail, and starting at the level of oxidative phosphorylation, the part which is really involved in the production of ATP. And this is really going back to Biochemistry 201, where we're looking at the generation of ATP through a series of protein complexes first that are involved in electron transport chain. They oxidize NADH and succinate, that's complex 1 and complex 2, feed electrons through the inner membrane through coenzyme Q over to complex 3, and electrons are then transferred to cytochrome C, often then onto complex 4, and oxygen is reduced to water. During this electron transport chain process, protons are pumped into the intermembrane space of mitochondria, creating a proton gradient. And this proton gradient is what is being used to through the complex 5 or the F1 FO ATPase to generate ATP through ADP and phosphate. That ATP is then sent out of the mitochondria in general, out into the cytosol and involved in then many metabolic and enzymatic processes. So this proton gradient that serves as a gradient to make the ATP is extremely important. It makes the membrane potential between the matrix and the intermembrane space, and that really drives the function of mitochondria. Without that membrane potential, ATP synthesis wouldn't occur, and also we'd have defects in many mitochondrial processes. So we need to have a very tight inner membrane to make sure the protons don't leak through. And so in order to get metabolites in and out across the inner membrane, we need these protein carriers to generate and or to carry them across, either into the matrix or into the intermembrane space. And even the proteins, all of those 99% of proteins that are found in mitochondria and need to go into the inner membrane or into the matrix, where there are very many different proteins, they need to be carried across through translocase components within the membranes as well, which are tightly coupled and prevent the release of protons during this process. So in order to make ATP and to make these very important complexes in mitochondria, which really contain very many subunits, for example, complex 1 contains 44 different proteins, we're going to need a whole machinery involved in their biogenesis. And we can have, with the defects in biogenesis, defects in the respiratory chain and the ATPase. And of course, we can have defects directly, and mutations directly in the subunits that are encoding the proteins that make up these complexes. But we can also have mutations in subunits or proteins that are involved in the biogenesis of these proteins. And I'll just talk about a few of these in a bit more detail now. For example, if we look at the oxidative phosphorylation machinery, where we've got complex 1, 2, 3, and 4, and complexes 1, 3, and 4 are actually known to exist in a so-called supercomplex within the inner membrane. These proteins these complexes are rather large. Complex 1 contains 37 nuclear DNA encoded subunits and 7 mitochondrial encoded subunits that need to be integrated together into this complex. And we have many others here as well. So we can have mutations in nuclear genes that encode many of these subunits that cause mutations in them that mean that these subunits do not function well. They may not function well such that when they get imported in the mitochondria, they don't assemble with the other complex 1 subunits, and causes complex 1 to be unstable, or it might cause enzymatic defects as well. So it's known that around about 32 genes directly encoding subunits of the oxidative phosphorylation machinery can cause mitochondrial disease. If we look at complex 1 in a little bit more detail, and this is just because we work on this a lot, because complex 1 defects are the most common defect in the oxidative phosphorylation machinery causing mitochondrial disease. Complex 1 is a beast of a machine containing 44 different proteins. It's a megadalton in size, and it contains a large protein complex arm sitting in the inner membrane of mitochondria and another arm sitting in the mitochondrial matrix. It's It's involved in this NADH dehydrogenase activity, where electrons are then transferred over to coenzyme Q, and in the process, protons are pumped across into the intermembrane space. Now, this is a conserved enzyme in that this complex is actually also found in bacteria, the origin of mitochondria. Yet, the core subunits of bacteria and our human mitochondrial complex 1 are basically the same. We've got 14 of those that are still in bacteria, and they're still in our human complex 1. But we've evolved to have extra subunits on top of this. We've got an extra, besides the 14 conserved core subunits, 7 encoded by mitochondrial DNA. They're actually very hydrophobic proteins that are integrated into the membrane of complex 1. We've also got 7 encoded by nuclear DNA that are imported and need to be assembled. They're the core enzymatic proteins. But we've got an extra 30 nuclear encoded accessory subunits that stick onto this complex. We don't actually know, or we haven't known clearly what their function is, although we know that mutations in a number of these accessory subunits have been found to cause mitochondrial disease. We in our lab have actually now knocked out the 30 accessory subunits in individual cell lines and looked at the defects occurring to complex 1, and almost all of these, loss of them, cause a total loss of complex 1 activity or function. There's only about 2 or 3 which sit right up towards this end of the complex, which seem to be redundant. While we have evolved a more elaborate machine, these subunits that are actually acting to sort of pepper over the complex and potentially stabilize it or prevent extra reactive oxygen species are still absolutely crucial for complex 1 function. And we've also looked at complex 1 assembly because it's a massive complex, 44 different subunits coming together at the end. And we've found, and others have found as well, that the mitochondrial encoded subunits are membrane subunits that are integrated into the membrane and form discrete modules, which then recruit extra nuclear encoded subunits which come into the mitochondrial matrix on the membrane and make these modules of complex 1, the building blocks. And these building blocks are then brought together and eventually build up the complex 1. And we know through this process that these building blocks actually require not only the subunits themselves, but also many different assembly factors to build it. Not just complex 1 requires this, but complex 4 requires this, complex 2, complex 3, and complex 5. They all need different assembly factors. And for example, in complex 1, there's over 12 complex 1 assembly factors so far identified that are involved in stabilizing these assembly modules, or actually creating these assembly modules in the first place in order to build up complex 1. And we know that many of these mutations in these assembly factor genes, which are nuclear coded again, cause defects in complex 1. So besides assembly factors that are coded by nuclear genes that come into mitochondria to help assemble these different assembly factors, we've got, of course, other protein complexes and other protein subunits involved in the biogenesis of the subunits that are important for making ATP. Proteins involved in import of the proteins directly into the mitochondria in the first place. We've got chaperones who are involved in folding these proteins and modifying these proteins. And also we've got proteases, which need to turn over defective subunits or even the complexes as they get old. And we need all of these, and mutations in all of these can cause defects in ATP synthesis. And at the moment, we've got around about 55 different genes that are involved where mutations actually cause defects in this machinery to cause defects in ATP synthesis and obviously disease. We also have another element, which is and these are a little bit of a different element to consider as well, is that we need a properly shaped mitochondria, and we need a proper population of mitochondria within our cells. And many different genes being now identified. I'm sure we're going to find many more, because we're still at the point, and I think David will mention this in a moment, around about 50 to 60. We know about 50 to 60% of the mutations that exist in patients with nuclear gene mutations. So there's many still to be identified. And we know that these genes are important also for the composition of mitochondria. For example, cardiolipin is a mitochondrial-specific lipid, and defects in cardiolipin synthase cause Barth syndrome, or classic mitochondrial disease, and it can cause defects in oxidative phosphorylation and the assembly of that as well. We also have genes involved in the dynamics of mitochondria to keep these mitochondria healthy and distributed and inherited amongst cells, and even to be turned over when the mitochondria get old. So if we look at differentiated cells and we see mitochondria and neurons, we know that mitochondria need to traffic up the axonal processes, and they need to be brought back down as well in order to mix and provide ATP to the ends of where the synapses are. So we've got to have proper distribution of mitochondria. We also have, in differentiated cells, mitochondria need to be organised really cleanly and to the needs of the cell. And if we look at a sperm, for example, the mitochondria in a sperm are actually wrapped around the flagella just under the head containing the paternal DNA. And it's wrapped around this flagella to help provide ATP for the swimming motion. And in muscle, mitochondria are stacked really neatly next to the myofibrils to provide ATP for the contraction processes that are important there as well. So we've got different ways mitochondria are arranged in different differentiated cells. And just to note that we've got machineries involved in distributing and causing mitochondria to change in the way they appear. And if we just look in a cultured cell, we can see, and here we've got a fluorescently labelled protein inside mitochondria that are orange, so we can just differentiate these. We can see that mitochondria actually divide. They break up into three, in this case it's 3 different bits. Mitochondria need to divide because mitochondria are inherited by cells that during division. We also need mitochondrial fission, as I'll show in a minute, for quality control mechanisms as well. But we also have fusion of mitochondria, which is a little bit more complicated. But this mitochondria here comes and kisses this mitochondria, and it moves some of this orange material within the mitochondria into this mitochondria as well before it breaks apart. You can see it here. There's a fusion event occurring. So it's a mixing of the material inside mitochondria in order to uniform the population of mitochondria within the whole cell. And that's an important element to make sure that, for example, the products of mitochondrial DNA are shared within and amongst all of the population of mitochondria. So mitochondrial fission is not only important for transfer of mitochondria between newly dividing cells, it's also been suggested to be a mechanism for quality control, such that when mitochondria can undergo a division step, mitochondria that divide may actually form 2 separate populations. A more sick mitochondria that has a lower membrane potential and unable to make as much ATP versus another one that may be healthy. Now, this unhealthy mitochondria may have the ability to fuse back and be repaired, or it has the ability to be recognized by the cell's machinery to turn it over. It has lower membrane potential, and there's a protein called PINK1 that actually becomes exposed on the surface of sick mitochondria and becomes a signal for the cellular machinery to be involved in turning or encapsulating this mitochondria within an autophagosome, so that they're sent off to the lysosome so that a more defective mitochondria can be degraded. So that's a way of potentially creating and maintaining more healthy mitochondria. So we've also got, my picture of mitochondrial DNA has disappeared, but it doesn't really matter. Fortunately, John showed a picture of mitochondrial DNA before. But obviously, we're looking much today about mitochondrial DNA, which is a circular piece of DNA encoded by 37 different genes. It has a light and a heavy strand, and there are multiple copies of mitochondrial DNA within cells and within mitochondria. We have hundreds to hundreds of thousands, depending on the cell type that we have of mitochondrial DNA. And mitochondrial DNA is actually packaged in the nucleoids within the mitochondria, and there's 1 to 2 copies of mitochondrial DNA are predicted to be involved in and packaged in these nucleoids with extra proteins. And it's thought that the mitochondrial DNA repair mechanisms are actually not as efficient as the nuclear genome's repair mechanisms, potentially because the mitochondrial DNA is obviously less packaged, and the enzymes involved in repair don't have that fidelity that the nuclear genome has. So we have the ability, and we have cells that contain mixed populations of normal and mutant mitochondrial DNA within our cells, and that can create a situation called heteroplasmy. We have too much mutant mitochondrial DNA that can drive defects within cells and show defects in ATP synthesis, and obviously if that's in the tissue, that can then be exposed as a real tissue defect within humans. And I'm sure we'll hear about this a little bit later on, but we have obviously inherited mitochondrial disorders coming through the maternal line, and the mitochondrial disease can be shown from birth to older age, and we can also accumulate mitochondrial DNA mutations with age, potentially due to reactive oxygen species damage as well. So we have mutations that are found in almost all of the genes of mitochondrial DNA, 12 or 13 oxphos subunit genes, 22 tRNA genes, and one of the 2 ribosomal RNA genes that are involved in this. And they obviously are causing defects then in the oxidative phosphorylation process as well. We also have, besides these mitochondrial genes that are defective, we have nuclear genes that are involved in the maintenance of the mitochondrial DNA, for example, the replication into making more nucleoids and making sure that the mitochondrial DNA is transferred into new mitochondria. And we have transcription proteins involved in transcription, all encoded by nuclear genes. RNA is processed by nuclear genes, so proteins are imported into mitochondria and process them. tRNA synthetases are found, which cause mutations of which cause disease. And we also have ribosomal subunits, which are built in with ribosomal RNA and proteins involved in assembly of the ribosome to actually make those 13 polypeptides that are left over within mitochondria. So this is my last slide, and I think it's probably about right time, just to indicate that we'll hear more about this, but mitochondrial disease really affects about 1 in 5,000 people. We have mutations in a variety of different genes, both nuclear and mitochondria. And this can obviously impair and affect many different organs, depending on the defect and the mutation that arises, and it can occur at any age. And with that, thank you there.
Thanks very much, Mark. That's a great introduction to mitochondria. Has anyone got any questions?
So, like the kissing.
Yes.
Does that, so in all sites we see mitochondria just assuming fusion at the same time? I think they would kiss.
I would think that they would kiss. So it could be a transient fusion, because they do look like nice little dots separated from one another. And it's going to be important that you're going to have trafficking of the mitochondria, but there's no reason why you shouldn't suggest that you got fusion, unless the fusion machinery are inactive. Okay, but for all intents and purposes, they should be. I don't know if anyone's really looked at this in detail.
Because, I mean, if they swap pieces of mitochondrial DNA,
Yes.
because one school of thought would be that they exist as individual entities, because that's a kind of quality control mechanism. So you're not swapping. So that if there's bad DNA in a single mitochondria, it's on its own and it'll get wiped out.
Yeah.
So, but if they were sort of kissing,
Yeah, but if anything, it has to be a transient interaction, a transient fusion, and it could be just the outer membrane fuses and not the inner membrane. But generally, we've seen that we get both. We get both outer membrane, inner membrane fusing, mixing, and then you get a division. Sometimes they just fuse and stay fused as well.
Yeah, we've looked pretty hard to try and find whether mitochondria do form networks at all in oocytes during growth. I mean, they're obviously undergoing a dramatic amount of biogenesis and fission when they go from small eggs to very large mature eggs. But they really do seem to stay as independent entities. That's an interesting, I think it's a very interesting difference between the cell lines and the cells and oocytes. It's morphology is really remarkably is very different. And as you showed in your slide, different cell types have very different
Absolutely.
mitochondrial structures.
Absolutely.
Any further questions? Okay, great. Well, thanks very much, Mike. We'll